All right, hello everybody. I'd like to introduce to you David Kong. He is an amazing guy. He's working in the Media Lab currently, but I met him when I was trying to learn something about online biotech. And it turned out in the basement of his house, he had a sort of a thing where he was teaching all of us idiots how to do biotech. Yeah. So David has gone along and thought hard about how to build a community that could start to do biotech. These are more common in areas where you have maker spaces, electronic maker spaces, and so on and so forth. David is trying to do this in a new area. So here, what we can see by his activities is sort of how this practice of distributed innovation amongst users is spreading. Right, David? So
Yes.
David is marvelous, and you want to carry on from there?
Sure. Well, I will return that and say, Eric, you are marvelous. I first met Eric, I guess it's been a number of years ago now. I think it was through Kareem how I met you, Eric. But yeah, it's really just been a thrill to be a part of this larger innovation community and thinking a lot about open innovation, user-driven innovation. Very briefly, my background, I'm a synthetic biologist by training. I'll talk quite a bit about what that means and in the talk itself, but I'm kind of an MIT lifer. I did my undergraduate, my master's, my PhD, my postdoc, then I helped to lead a synthetic biology center at Lincoln Lab for a handful of years before joining the Media Lab, where I direct a research group there now. And my father was a professor at MIT as well, so I'm literally like, I have not cut the umbilical cord. I'm one of those. But it's been such a thrill to engage with Eric, and part of it for myself, again, my background, because I was a technical person working in the technical field, I was working on chips and devices and instruments, which I'll talk a little bit more about in a moment. But I was also a community organizer and a social justice activist. So my interests really kind of started to merge together, I want to say around 10 years ago, when this larger field of what's called do-it-yourself biology started to emerge. And I'll talk in depth about that in a moment. But to me, I think it was really a connection between this social dynamic of who ultimately gets to benefit from these technologies, who ultimately gets to shape them, who has the agency, who gets to participate. Being somebody that was working at the forefront of this field at MIT, where it's basically all the experts, right? I had a certain kind of sensation about it. And then when DIY Bio emerged, I started to realize, wow, this actually starts to bring together my social interests and my community-driven interests with my technical interests. And that really fundamentally changed my career. So for the past 10 years or so, I've still continued very much on technological development, but have been really thinking deeply about how do we share things? How do we open source things? How do we get as much diverse participation as possible? And I think through, working with folks like Eric, and folks like Kareem, I've been able to think a lot about, from an innovation perspective, why, diversity is an important thing, for example. And it's basically given me a language and a structure to think about as we work on the technology side, why this effort is important. And all of that to say, I am very much as well a practitioner and a prototyper and a hacker by training and by ethos in a way. So much of the work that we're trying to accomplish, it's all experiments. Like we're all trying different types of experiments at different scales, both technical experiments and social experiments. And so, it's really exciting to be here to speak with you all, because I think, I'm also excited to learn and think about how we can also shape our movement and shape our work in the best way possible. So, so that's just some introductory remarks, and I can dive into the talk. So let me just share the screen and make sure that this works well. Okay, are you all seeing, if I full screen this now, are you all seeing the screen here?
Yeah.
Okay, great. Okay, so first, just a little shout out to my group at the Media Lab. So again, I do work that is in an academic context, but then also I do a lot of this global distributed work, which I'll talk more about in a moment. And so just a few key terms as we get into this today. So part of the work that happens in the life sciences and in engineering I kind of break this out into two areas because we think about both science and engineering. So things like molecular biology, microbiology, cell biology, that's understanding the fundamental science of either cells or certain types of biomolecules, et cetera. But then there's engineering, so actually building and making stuff, right? So synthetic biology, which is kind of the field that I was really born into in the early 2000s at MIT, this is fundamentally about engineering. This is fundamentally about how do we build and make stuff. And so down here, I've put a bunch of other terms that often are used and sometimes mean the same things, but can be very confusing. I think, recently, some of you may have seen, for example, the Netflix series Unnatural Selection, which talked a lot about biohacking. There's a Showtime movie that's coming out soon that I've been a part of that also, it's called Citizen Bio is the name of that documentary that's coming out. So increasingly, there's been a lot more media attention around what happens with both the life sciences and bioengineering when it's happening outside of the traditional context, when it's happening outside of an academic institution or an industrial laboratory. What happens when people start making stuff in their garages or set up independent community labs and grassroots structures? So do-it-yourself biology is something you hear about. Biohacking is another term that's become popularized quite a bit, although I think some of you also may have heard, within the past couple of years, there were some pretty high-profile stunts where people were like injecting them on stage, injecting themselves on stage at conferences with like, herpes vaccines and other things. And so biohacking, I think, as a term has maybe gotten a bunch of negative publicity because of some of those actors. But as you all know, at MIT, hacking is still something that's reasonably well celebrated. And so biohacking is kind of a complicated term. Biomaking, I think, is one of the least controversial terms. At MIT for a number of years, I was really an advocate of MIT setting up some type of a community laboratory space, right? There are maker spaces all across campus at MIT, but there was no place where if you were a smart student that had a cool idea around bio that you could go do and a place you could go and execute a project that did not exist in the auspices of a research laboratory, right? Where you had to get yes from certain folks. And so when the students were first organizing that, they were calling it a biohacker space. And they got so much pushback from different people all around MIT. And I suggested, if you call it a biomaker space, I think that'll be a better term. And so then all of a sudden it took off, and now there actually is a biomaker space on campus. And Marty Culpepper, again, who's the maker czar at MIT, has been really doing a fantastic job, I think, of trying to pull this all together. But a term that I've really been trying to push is, I welcome your feedback on this term as well, is maybe there's something that's called community bio, that is not about do-it-yourself do-it-yourself has, I think, an ethic to it, but also I think just built into its word, its ideas that, oh, we're doing this ourselves. Whereas I think now there is an emergence of global networks. And to me, that's, I think, one of the really exciting areas is thinking about how you could get diverse populations from across the world collaborating and working together. And that's a big part of the movement building work that we've been doing that I'll talk about later. So the first part of my talk is really technical, explaining what synthetic biology is. I'm going to go through this quickly. I've got a lot of slides here, so I'm just going to go through it very quickly, but please stop me, interrupt me at any time. And just to say that in general, I really enjoy discussion and conversation. So just at any point, please interrupt me. So I wanted to start by saying first, we think about synthetic biology and bioengineering, but actually humankind has been hacking the living world for thousands of years, right? Going back first to agriculture, to domesticating animals and so on. There's been this idea that humans are influencing and manipulating the living world. And so the era, of course, that we're in right now, you might call this the synthetic biology era, but really I think it's a molecular era where we're now actually able to provide direct edits and modifications into DNA. And so this really started, potentially as early as the late 60s and into the 70s, but really the recombinant DNA era of basically taking DNA that's existed in nature and then using a set of tools called enzymes, which are basically proteins that can then edit that DNA, that can copy it, can paste it, and so on. And so this was a really powerful technology that emerged. But in the area of synthetic biology, where we are now, perhaps the most powerful technology of this field is something called DNA synthesis, which is literally starting with bottles of the individual DNA bases, A, G, Cs, and Ts. And you have this machine called the DNA synthesizer that can basically string together these arbitrary sequences and create new molecules that have never before existed in nature, okay? And so just kind of wrap your minds around that for a second, that there's literally a printer. There's like a device that you can input a bunch of code into, if you want to use that metaphor, which I'll talk a little bit about in a moment, and hit print on a machine, and out comes this new DNA sequence, right? So this is a really transformative technology, and it is really foundational to I would say, modern biotechnology and the life sciences as a whole. And so over the past, I would say 15 years or so, 20 years, we've seen DNA synthesis proliferate where you can now manufacture arbitrary molecules of DNA up to sizes including those of whole bacterial genomes. So going up to megabases, so hundreds of thousands of base pairs to millions of base pairs. So a lot of the work that is in kind of modern synthetic biology is in the thousands of bases, so individual genes to genetic circuits, which are, say tens of thousands of base pairs. And what I want to kind of emphasize here is that you'll notice there's a really interesting metaphor that's emerging. So you'll see genetic circuits here. And in the next slide, I'll talk a little bit about a very seminal paper from 2000 on a ring oscillator for biology. Okay, so synthetic biology in many ways was this very interesting experiment of a metaphor, right? Can we actually use metaphors from engineering, particularly from electrical engineering and computer science, and map that onto biology? Right? And so this paper in 2000 was the first demonstration of a ring oscillator, which is a fundamental component in electronics, but doing it in a biological circuit, quote unquote, where you basically, this circle over here is basically a plasmid, it's called. There's a bunch of DNA with a bunch of different proteins that expressed on it. I won't get into the gory, nerdy guts of it, but the short of it is that when you inject this plasmid into a cell and you have it replicate over time, there's a fluorescent protein that gets expressed in a circular way or in an oscillatory manner that behaves just like a ring oscillator in electronics. Okay, so this paper in 20 years ago was like, people all of a sudden were like, holy crap, like you can actually have a living system that has evolved over billions of years and actually play with it in a way that you might with in an engineering perspective. And so the other thing I'll mention here, I don't have slides on this, I won't get into it deeply, but there are so many other engineering principles and paradigms that people are trying to bring into synthetic biology. So things like having very well characterized, standardized parts, right? So if you got a parts kit that you can build from, I've got defined inputs and outputs for a system, and I can stitch them together to make a cool new, kind of biological device. And so this circuit that you're seeing here, you could imagine decomposing that into these quote unquote biological parts that could be, I'm going to say a bunch of technical words, but like promoters or terminators. So different DNA elements that we're aware of that could be strung together and built into a system. Okay, and so this is how you start getting these like very complex looking circuits that actually have behavior like digital Boolean logic. So, and this again goes back to this larger metaphor of, is the organism something that is sort of like a computer? And is it something that where if you can engineer that DNA code, then you can then kind of get it to do something new and valuable. And so this is a picture from, Technology Review at MIT, kind of, which I think brings together the metaphor in a neat visual way, right? Like you've got this, a potentiostat and all of these other, kind of elements built into this bacteria. And so, all right, so now we've got this metaphor, we've got DNA synthesis. What are people doing with this tech? One of the huge modalities that you may have heard a little bit about is the idea of turning cells into factories to make stuff. Okay, so right now, literally in our bodies, cells are producing all kinds of compounds and molecules inside our guts. Very, very valuable short chain fatty acids, different types of neurotransmitters, all sorts of really valuable and interesting chemicals and compounds being made by our bodies and by the living world all the time. And so a big area of research is figuring out all of the metabolic pathways that are out there in nature and figuring out, can we actually engineer organisms and get them to make useful stuff for humans? So what are some of these useful things? So that, for example, chemicals and fuels, right? Literally some of the early demonstrations in synthetic biology were engineering bacteria to make a variety of different types of fuels and chemicals. One of the companies that started, that's now probably the biggest synthetic biology company in the world, is called Ginkgo Bioworks, which some of you may have heard of. I went to grad school with all of the founders, and literally it was scrounging for equipment in the hallways of MIT, and it actually kind of started out like a garage lab, to be honest. And what's actually even full circle more interesting is Ginkgo, after they became more successful, they were the place, if you were setting up your garage lab, where you would go to them and they would give you their kind of third or fourth generation equipment. So, in our community lab in Cambridge, which I'll talk a little bit more later, we've got all of this Ginkgo equipment that probably was part of some of the first equipment that we found, like, in the halls of MIT 15 years ago. So, so they very much had a hacker aesthetic to start with. And so anyways, I'll skip this one for now. I'll come back to that later. But also, cell-based therapies are another huge area. So engineering these organisms that you could actually install inside the body that could directly sense and respond to disease. So instead of taking a pill or a medicine, this engineered cell is actually the medicine and is actually directly responding to inflammation, for example, and then treating it directly. So there's a whole area called live therapeutics, which is a big thing that people are working on. Cellular agriculture is another huge field which some of you may be familiar with. So, how do you create animal products without animals? Can you grow these in laboratories and so on? So there's a whole kind of industry around this. New Harvest is another organization. This is another kind of neat one, which is, can you actually leverage these technologies to then produce products and materials, things like for fashion? So this is an example of a clothing that was made but secreted using bacteria, okay? I give all of these examples as well because there are user innovations of all of these, right? So this is, I'm showing you what's happening kind of in the industrial setting, but I'll talk more about how, regular folks from all around the world are engaging this as well. And so spider silk, that's another kind of, really cool one. One of the things that terrifies me is the idea of like a spider farm. But fortunately, you can actually get this silk just by fermenting it in a bioreactor. So this field has really exploded, and now billions of dollars a year are being put into this industry. And so kind of what I've shown you are products, some examples of products, but I'll just put this out here as another example of something that maybe is a little bit more profound and maybe even connects with part of what we're seeing right now with this current pandemic and COVID-19. Obviously, we've got a situation with the life sciences where things can self-replicate in labs, right? Like you can actually have stuff, and that's one of the properties that we want in the case of like a fermentation where you're trying to, manufacture a molecule. But this is a project or a paradigm that's been developed by my colleague at the Media Lab, Kevin Esvald, called gene drives, which is a specific application of CRISPR-Cas9 technology. So I think you may have heard about CRISPR, but CRISPR in short is a very precise genome editing technology. It's one of the, there are a number of these technologies, but CRISPR has gotten a lot of press because it is quite easy, relatively easy to use, and it's something that has been proliferated throughout labs in a very short period of time. And so gene drives are a particular application of CRISPR where the edit that you're trying to make in the genome, in this case, this would be, say, for a mosquito, the edit includes the CRISPR machinery itself. So in this case, for example, let's say you're trying to create a version of a mosquito that is resistant to malaria. Then you could have that trait and actually through sexual reproduction, because you've got the CRISPR machinery built into it, actually drive that trait through the wild population just through sexual reproduction. So after multiple rounds of sexual reproduction in nature, you end up with a population of mosquitoes in this case that all have that genetically engineered trait. So what we're saying ultimately is that there's very profound impact that this technology can have for the world, right? For our ecosystems and also for human health. So that's some of the tech. Now I'm going to get into some of the tools, and then I'll talk about how we're actually figuring out how to open source and get some of these tools out there. So again, my background, one of my areas of technical expertise is something called microfluidics. So I show you this as an example of a technology. This is a pipette patent from 1916. And then this is a modern pipette patent from 2012. And it's kind of stunning to me that this thing hasn't really changed very much, like 100 years, and we're still kind of manipulating fluid in a similar way. And if you go to a wet lab, actually, so much of what happens in a wet lab is actually moving around liquids. And you're not really sure what's going on. You get some measurement at the end of it. But one of the technologies that I work with very closely in the lab is something called microfluidics. So lab on a chip. I'll go through this very quick, but basically the size scales that we're talking about are very small. The dimension of the width of a human hair is around a typical size for one of these devices that we make. And so we're typically working with these very small cells, bacteria or human cells that could fit inside our devices. And so these chips, they're programmable. You can manipulate them with a computer and do all kinds of cool stuff with these devices in terms of mixing, manipulating fluids. But the whole idea is to basically take a lab and put it into a chip is kind of the idea. And so I mentioned to you earlier this DNA synthesizer, this kind of magic machine that kind of produce these synthetic DNA molecules and print them. And so part of my PhD thesis, also at the Media Lab, was building a microfluidic gene synthesizer, so building gene-length fragments. And then in recent years, we've been building these larger circuits using a bunch of open source hardware. So I'll pause here for a moment to give you an example of a device that we built with the intention of setting it up so that a global community could actually hack with it, work with it, and build their own devices and make remixes to it. So this is an example of a tool where you've got this Arduino kind of breadboard in the middle that's its brain and guts, and a series of kind of valves and a kind of standard PCB layout. And for us, we were very interested in kind of sharing these design files, and there's a larger infrastructure, which I'll talk about later, called Metafluidics, which is a big platform that we have for sharing all of these design files. So I would say, again, going back to Eric's original prompt, what are some of the strategies that we're working on? From a tool perspective, one question is, how do we make these tools more accessible, right? If you go to a traditional biotech lab right now, there's so much infrastructure that's required to operate and work in these laboratories. So one fundamental question is, how do we actually make the tools and technologies and the infrastructure necessary more accessible? And that accessibility could be from a cost perspective, an availability of parts perspective, and also critically an ease of use perspective, right? So for us, in our lab and the labs I've been a part of, a big part of it is tool development. What are the kind of tools that can enable other communities to come in and actually work with them, but critically also to play with them, right? We want you to ideally hack with the tool and build your own versions. And so there's some ways, again, I'm sure you've been learning about in class about how you can structure that better or worse. And so we've done that with varying success over the years, but this is just one example.
David, if I
yes.
feel free to punt this question if you're going to touch on it later.
No, please go ahead.
But I'm kind of curious for your lab, how you guys are thinking about like the ethics of distributing some of these tools.
Such a great question. Such a good question. So I think the first thing to say here is that, when you talk with folks that are quote unquote experts, I think people think about ethics like it doesn't apply to experts. Like it's like the experts are somehow totally ethical, and that the quote unquote users or the non-experts are not ethical. And one thing that I find is so fascinating is that, I find at least, this is more anecdotal, but in the grassroots communities and the community labs, which I'll talk about in a moment, kind of the hacker spaces, I feel like the discussions of ethics happen far more regularly and far more frequently than in my own academic training. So even in all my years at MIT, the number of times that I've actually been in conversations with technical experts to say, hey, is this the right thing to do or not? Not that those conversations don't happen, they do, but it's just an interesting kind of framing. And I think for myself, one thing that we've done a lot, and again, one of the big efforts I'll talk about in a moment is something called the Global Community BioSummit. It's an event that I organize every single year at the Media Lab at MIT. And we basically convene the global network of these community labs. We bring them all to MIT, and
David,
one of the
here.
Yes.
So one of the things you'll see here, I mean, Maria, next time we'll talk for a few minutes about sort of regular kind of like electronic and maker spaces. But what is happening here with David is something sort of much more fundamental in a way, or let's put it this way. Historically, we've all hacked electronically. However, many fewer people have hacked in a biology sense. So what David is trying to do here is quite remarkable. He's trying to build the basic set of tools that people dragged into the electronic maker spaces, the software maker spaces, because everybody knew them, and to create those tools and then make them user-friendly and then create the community. So it's really quite an extraordinary thing that he's working on.
Yeah, and to build on that, Eric, I think there's three dimensions about this. Three core ones, I would say. So one are the tools themselves, which we're talking a little bit about now, and I'll talk a little bit more about in a moment. Another part of it, though, is actually education, right? So that's a huge, huge part. So, just because you've got a bunch of tools, great, but what's the structure and how do I learn about it? And how do I actually ultimately, have some basic, what we like to call sort of DNA literacy, right? Something I think that's increasingly happened in part because of the pandemic. People are starting to understand a little bit more about like what a virus is, for example. So that is a really huge area, which is just education. The third part, though, is innovation, right? And innovation might be the most challenging part, which is how do you actually get, folks, and again, I think just as a question for you all to be thinking about, and I think there may be a discussion that breaks out a little bit later too, but when you have a system where you have very uneven distribution, I would say, and expertise, you have some folks that are coming in that may have some academic training or technical training, but some people that are coming in that may be very creative and bright, but have never been in a wet lab before, right? If you've got a very heterogeneous group of people with a variety of skill set, how do you best structure and coordinate them so that from a technical innovation perspective, you can end up with some real winning ideas, right? That I think is the big challenge that I still work with and grapple with and think a lot about. And so that might be something just from a principle perspective. This doesn't have to be in the life sciences, right? This is, I think, is a hopefully being abstract that out to other disciplines, right? But when you have this very heterogeneous distribution of skills, you know, how do you coordinate them well, right?
Yeah, yeah.
And I'm sure that's something you've been talking about in class. Yeah.
Yeah, this is marvelous because you see all this other stuff like in computing and so on has sort of grown from the grassroots and never did anybody think about it very hard about the underlying principles.
Yeah.
So what David is doing, and I'm really impressed as hell by what he's doing, is that he is saying, here's a new field, you know, and all of you in the management school and so on, and the engineering school should say, well, what he's really trying to do is think through how to make the conversion from centralized innovation in sort of MIT-like places to grassroots innovation. And it's fabulous. It's really what all of us will have to do in our particular areas if they have not already been converted. Sorry, David,
So,
that was a rant.
No, I love it. I love it. So I'll move quickly through some of the next material just to get into the hacker spaces, because I think that's part of what's really cool. So anyway, so you can do all kinds of cool stuff with these. Oh, sorry.
Before you do that, sorry.
Yeah, please, go ahead.
Dean has a question for you. Or Dean, if you want to jump.
David, is this okay if I ask a question, or would you like to?
Of course. No, of course. Please go ahead now. Yeah.
I'm very curious. Who are the kind of folks who are starting these community labs?
Yeah, such a great question.
From traditional academia, outside.
Yep. I would say it's a very big mix. Some of the earliest community labs that were set up, so there are two kind of founding labs in the United States. One was called GenSpace and the other was called BioCurious. GenSpace, both of them started up as meetups, right? They were literally just meetups. And again, I think a lot of it, in my view, was a reaction to synthetic biology. So synthetic biology started to emerge, was getting a lot of really interesting publicity and press. And for that field, it was pretty positive press, I would say. And so I think a lot of the early quote unquote citizens that were getting together, GenSpace was organized by a journalist. There was a journalist, Dan Grushkin, who founded it, who's one of the founders, organized it and just put out a thing on Meetup and said, hey, you know, who wants to get together, right? So a lot of it, I think, if you were to kind of analyze the demographics, I think there were also a number of computer scientists, I think, computer scientists and engineers that were not in biology that were getting into it. And that actually, I think, mirrored well the founding of the field of synthetic biology as a whole, which was founded by non-biologists, which is another one of the points I'll mention a little bit later. But you know, Tom Knight was kind of a legendary computer scientist and was like, hey, you know, why can't we program cells like we program computers, right? Drew Endy, another one of the kind of fathers of the field, was a civil engineer. He was a guy that built bridges and designed the built world and said, why can't I apply engineering principles like I do in my field to biology? So this to me has always been a really wonderful anecdote around the power of diversity and having these diverse perspectives in a field. And so, so I think the community labs, again, started with a bunch of engineers, creatives that were not necessarily biologists. And I think what we've seen in the past like 5 years to 10 years has been the emergence of artists, designers, other folks that are really coming into the field and making a big impact. There's a whole new institution that's been set up by Dan Grushkin, the guy that founded GenSpace, called the Biodesign Challenge, which is all about engaging designers. So that's been it's a really wonderful competition that happens every year. I've been a part of that as a judge for many years. And so this, I think, is one of the hallmarks of what I would call community bio, which is really bringing in these networks of diverse technical and creative backgrounds, which I think has been consistent with the field since the start. But great question.
Thanks very much.
Okay, so I'm going to move quickly through this stuff, this part. So, you know, we've made these devices, and you know, you can use these circuits to look at cancer. Protein engineering is a big field as well. You know, if you've got toxic spills in the environment, how could you get a good enzyme for that? Obviously, for something like COVID right now too, you know, finding the right types of therapies, protein therapies, gene therapies, and so on. So these are all examples of different chips that you could use. One area that I find really exciting is the microbiome. Some of you may have heard about that, you know, the trillions of microorganisms that inhabit the body. I will skip over this other than to say that, you know, you may have heard about fecal matter transplants, which is one of the kind of more profound engineering ways, in a way, that you can work with the microbiome, where you transplant the fecal matter of a healthy patient into a sick patient, one that often has a disease like Clostridium difficile, which is a common infection you can get in a hospital. I bring this up in part to highlight one of my friends and colleagues, in a way, Josiah Zyner, who's one of the guys that's gotten a lot of negative publicity. He is one of the guys that injected himself in the arm with CRISPR at a conference. He also, before all of that happened, was kind of famous for doing self-administered fecal matter transplants. So he got a lot of publicity because he himself had some gastrointestinal issues. And then, you know, again, his whole, he likes to kind of describe himself as the mad pirate king of biohackers. Okay, so there's a certain kind of strand of what you might, if you think about maybe community bio as like an umbrella term, you know, Josiah is definitely kind of the leader of the biohacking movement. And I'm sure as you see in other types of user communities as well, there are many factions, right? Going back to the ethics question that somebody asked earlier, Josiah is a person that hates ethics. He hates bioethicists, and he's kind of like a hardcore libertarian that believes that like nobody should stop me from doing whatever I want to do as long as I'm not hurting other people. And I probably relative to Josiah am more moderate. So you know, in answering the question from earlier, one thing we've done at the Bio Summit is we've been facilitating these ethics processes. So we actually last year set up a whole co-creation of a community ethics document, which I'll talk about later on. But for us, I think one of I would say the philosophies that I've really been embracing is this notion of co-creation, right? Getting the communities that are the active participants and the agents to actually help to co-create the culture and the structure through which we actually are going to do that work, right? So I think Josiah has really captivated the minds of a lot more of like kind of the rogue biohacker, whereas I think we've been trying to do it in a more structured movement style way when we think about our user community. And so anyways, I'll skip through these parts. Okay, so all that to say, you know, we've made a bunch of cool tools, but how do we make these tools accessible? Okay, so this is kind of the getting into the open source aspects of it. So many of the tools that I'm showing here, actually, in fact, all of these tools, the Bento Lab, the Amino Lab, OpenTrons, and Gaudi, these all came from hackerspaces. These all came from basically garage or community biolabs. And so all of these tools were developed in a way to get at that question of accessibility, right? As I've mentioned before, you know, Ginkgo Bioworks, you know, all of these sophisticated biotech companies, the amount of capital that's required to get a basic kind of life sciences experiment going, if you would use kind of traditional industry or academic routes, is really significant, right? You need a lot of equipment, you need a lot of infrastructure, there's something called the cold chain, which again, if you know, so much of biological reagents, you need frozen, either at minus 20 degrees, sometimes minus 80 degrees, right? These are very expensive like pieces of equipment. And so one thing that happened within the hacker communities was they said, well, what can we do to make these tools cheaper and easier and more accessible? So Bento Lab, Amino Labs, these are all examples of basically desktop biolabs. And this is very much borrowed, I think, you know, in metaphor or you know, kind of story from personal computing, right? You know, similar time, you've got mainframes and all these other systems, and then, you know, a bunch of people said, hey, like, why can't we build personal computers? And I think that was part of the ethos that drove a lot of the early folks that were building these early tools. OpenTrons is a particularly cool one. This is a liquid handling robot. So normally liquid handling machines in industry are like 30,000, 40,000, or even more expensive, hundreds of thousands of dollars. And they basically built a commodity liquid handling robot that could cost several $1,000s. And this was actually invented by a social justice activist. It was a dude who had no biology background at all, my friend Will Canine, and was basically, you know, he was kind of a Marxist. He was like, the people need to control the means of production. And so if in biolabs they're using these giant liquid handling tools, well, I'm going to make like the cheapest one I can figure out and give it to everybody I can and open source the design files. Now, Will, it turns out his company now is like a very successful VC, like Silicon Valley backed startup. And it's, you know, they've raised millions of dollars, and actually they're playing a really big role in the COVID response right now, sending out armies of these robots to try and get labs ramped up so they can do COVID testing. But this is all just examples, I think, of some of the cool tools and kits that started emerging from this ecosystem of laboratories. Gaudi Labs is another really cool one. It's a Swiss community biolab where they basically, using a laser cutter and acrylic, figured out how to build almost a huge number of tools that you find in a wet lab. And so, as Eric was saying earlier, there's been this really interesting kind of dynamic between centralized versus decentralized, right? So we talked about DNA synthesis. And so there are all of these companies that you can actually go and order a sequence and they'll send it to you in the mail. You know, Kilobase is an example of another product that came out of a community lab in Europe where they were trying to build like a little chip-based DNA synthesizer. So, and so we're seeing that also, you know, with sequencing too. So there's reading DNA and writing DNA. So sequencing is reading DNA, synthesis is writing DNA. And there's also this kind of interesting interplay between the centralized and the decentralized. And so, so again, we built a whole bunch of tools. This is a neat example. When I first joined the lab, I had a bunch of undergrads and a couple high school students, and I challenged them. I said, hey, here's $1,000, go build yourself a molecular biology lab. And they basically got to work, and over the course of the summer, built a bunch of useful tools. So here's a couple examples, microscope, a centrifuge, a thermocycler, which is something you can use to modulate temperature, which is a very critical part of the life sciences. Electrophoresis, which is how you can separate different types of biomolecules. Transilluminator, something used to visualize them. This is a picture from inside our community lab, and this is George Church. If you don't know George Church, George is kind of the modern Charles Darwin in some ways of biology. And so, George has been a great friend and collaborator, and also a very big believer in these user communities, I would say. This is an example quickly of a tool that we built in our lab to basically facilitate this process of injecting DNA into cells. The cool thing I'll say about this is that it was invented by hacking fly swatters. So my student Teja, who is from India, was thinking to himself, what is like a globally available, source of high voltage, basically. And so he came up with this idea that, fly swatters are globally available. They're in almost every single country. They're very low cost. And then he basically, using all these very common materials, made a very low cost electroporation system, which are typically thousands of dollars, and a core tool in the life sciences, but normally not very accessible at all. So again, this is an example where we built a cool tool. We're in the process of putting this out to the world so that other people can then, both build their own versions, but then critically enable exploration, right? We want people to explore, use a fundamental tool like this to look and kind of survey the bacterial world, which is, this vast world. When you think about the scientific perspective, the vast majority of microbes out there in the world are unknown. They're called uncultured organisms. It's just, in the life sciences, there's like junk DNA, which basically just means we don't know what the DNA is. And in the microbial world, it's like the vast majority of microbes that we find in the subway system, in the ocean, in dirt, in our bodies, we don't even know what these things are. So part of, for us, if you think about what academic labs or industry labs could do, you can only get at so many just because there's only so many labs. So if there's a way to empower users, for example, to actually go out there and get the science done, that could be really marvelous and really, really amazing. So this is something that, we've been really excited about. I guess the other comment I'll make here too, and Eric and others, I'm curious about your perspective, but I think there's a really interesting question of what's the balance between the kind of grassroots user and then the expert, right? What is that relationship and what should it be, right? I think earlier on, I had this idea that why do we need experts, like the users could kind of figure out anything? Over time, I've been thinking to myself more that that the role of experts, that there is a role for experts, but that you have to think very carefully when you're thinking about whatever structure or problem it is, how to incorporate them. So I'll give you one example, just what's happened with COVID, right? Immediately after COVID-19 kind of became a pandemic, and it was clear how disruptive this was, you started to see the emergence of all of these different folks from around the world that wanted to engage in the problem. They wanted to help, they wanted to work on the problem in some way. And I would say there were some activities that were incredibly unstructured. There was literally like a Facebook group of like 20,000 people being like, hey, I want to help. How do I do stuff? And with very little quote unquote expert guidance or people that really understood stuff about diagnostics or how you could set up a trial or whatever it might be. And so I think in watching that process, one thing that's occurred to me is that actually experts can be very valuable in structuring problems, right? Experts can be very useful to say, hey, look, you want to do something complicated like build a diagnostic or make a ventilator. That was something that, again, was a very big deal in the broader maker community, which some of you may have heard about. But ventilators are really hard to make. I mean, they're very complex with lots and lots of different kind of complex elements to them. So one potential role where experts could play a role is actually helping to structure the problem to say, hey, there's a part of this problem that's informatics. And so this is where we need help here. There's a part of the problem that's about hardware and we need help here. So that may be a useful modality. But again, I think this is just an interesting discussion point. I don't know if Eric, you want to talk about this for a moment, but to me, this is one of the things that I struggle with or think a lot about is, because I straddle both worlds, right? I help to run a community lab, but I also am nominally a quote unquote expert. I've got the fancy degrees and I run a lab at MIT. So people look at me and they may think I know something that I'm talking about. I think it at least, but Eric, yeah, I'm curious what you think about this, because this is a very important topic to me as well.
Well, it's a really interesting one. I think you have bridged a couple of things. The first thing is that when we study where user innovations come from, they tend to come from people who on their own have expertise, right? So when you see a crowdsourcing thing or something else, it's people joining in who have expertise. And when you search for innovations in the world, user innovations, what you're doing is you're screening and finding the stuff that for some reason people did on their own because they needed it, right?
Right.
Then very often what you find, like the origins that you mentioned of synthetic biology, is that there are people with expertise you wouldn't have expected, right? Who come in and say, hey, I can do this, right? But it's not the same as people who say, I really don't know what I'm doing, but I'd like to help. And so that's why I've been talking with you, or thinking that we could chat about. I don't know what you think, but thinking
love to, of course.
that you've got two different roles here. One is you've got people with sources of expertise, and you can sort of bring them together by this machinery and so on. And the other kind of mode is this, let's all get together and try to help people who don't have expertise contribute.
Right.
And I think they're two very separate things.
Right. Yeah, I think that's actually a really great articulation. So the second part, I think, is much more about the education and learning part, right? Folks that just don't know and just want to understand, like, I'm reading in the news about CRISPR, I want to understand what this is. And so part of what's been so valuable, in my view, about the community lab kind of ecosystem, primarily the labs, almost every single one of those labs has a very strong educational component, right? MIT, of course, does some stuff around open learning and so on, but there's no lab, as far as I know, not even our lab, that you can walk into as a member of the public and try to understand what's going on, right? So the community labs that have been built out, this larger network, I think it's been so critical for establishing this type of learning environment, hands-on learning environment in particular, workshops. You can go and actually do wet lab work, right?
So I'm just saying they're two separate functions, David.
Yes.
Right? One function is that people who have unusual kinds of expertise from around the world can pull that together. And the other is an educational function. And I think you mix them at your peril, because it's not clear how long those people who are really experts in trying to do what they're doing want to be in the educational role. They're they're an innovation role and an education role.
Great question. I think we got a hand raised. Harold?
Yeah, I'm just wondering about complementary expertise. You've worked with Kareem. Kareem introduced me to Innocentive, and the best example I have is Lilly looking for an ability to manufacture a polymer that's used in agriculture, and the answer comes in 3 days from a petroleum engineer in Kazakhstan. So I'm wondering about the role of complementary expertise in community workshop.
Yeah, so that's actually a perfect segue into some of this next stuff. So this is a class that I taught, I think about 5 years ago now. And there are a couple of folks in this photograph that I wanted to highlight. So this guy in the red all the way on the left, his name is Will Patrick. Will was a designer that had never set foot in a biology lab before until this class. And he, within a couple of years, he got his master's at the Media Lab. Within a couple of years is now the CEO of his own company called Culture Biosciences. And they're basically the hottest bioreactor company in Silicon Valley. They are just so, they've raised a huge amount of money. They're doing fantastically. And part of, I think, why Will was successful is because of his design background, he really knew how to make products that people could interface with, right? And so this, again, I was mentioning earlier, this notion of kind of bio designers and bio artists starting to get into the field. I think that to me is one of the most exciting areas, is because now you're bringing in sort of what Eric was saying, you're bringing in people with actually deep expertise. They've got deep creative or technical ability in a discipline, but now they're bringing that unique perspective in, looking at your field and then seeing things that you don't see because you're used to a particular dogma or a particular perspective, right? And so for me, one of kind of my meta innovation goals in some ways is this kind of social experiment. Okay, if we start bringing in even more, and you can think of diversity beyond just art and design too. I mean, there's so many other ways to think about diversity. So when you bring that all together, how do you structure that mix? And so this is, I think, is Eric, what you were getting at earlier, which is the nuances of how do you bring these groups together so that certain folks feel really excited and motivated, but don't feel like, oh, I'm just being brought down because, you know, whatever, the people over here want to do this and I want to do that, right? And so these, I think, are some of the nuanced challenges that we face in structuring these different formats through which people can engage in innovation, right?
Right. I agree. I mean, I'd raise one point, which is nobody's saying a person who wants to come in is not a worthy person. But, you know, if it's like anything else, if you want to come into a community with expertise in something, there's a distinction between the teaching role and the getting something done role, right?
Yeah. Yeah.
So this is sort of what I'm thinking with respect to your thinking. It's,
yeah.
you know, I used to go crazy when I was in Denmark, and you know, we would put together this group of people who were really good innovators with respect to user innovation in some area, right? The Danes would say, but this is not democratic. And so they'd bring in a horde of people who happened to be wandering by, and those people were then given Post-it notes, and they'd post it all over the wall, and then they left feeling satisfied somehow. But it never struck me as a sensible sort of way to tell people how to interact with the process. So
yeah.
Anyway, it's
no, go ahead.
a separate conversation. But
okay.
Yeah, but keep going. I mean, this is very exciting. Sorry.
Okay, no worries at all. Yeah, and I'll get into a little bit some more of the educational institutions that we've built up to try to get at education and the distinction between that and the innovation part. Okay, so anyways, this is just some of the stuff we did in the class. I'll pause here though, just to give an example, right? Like these are devices that again, Will Patrick made, and you know, as a designer coming in without any technical ability, he made some of the most interesting structures that I'd ever seen. I'm a microfluidics guy, and so, you know, I've designed so many of these devices, and they probably looked more like the second row, the stuff that I had made. And Will made all this stuff in the third row that I was just like, what is this dude doing? Like, this is like, I'd never seen stuff quite like that before. And which again, I think just goes back to and hopefully reinforces the idea of you know, having these very diverse technical and creative perspectives in the field and how that can lead to some really cool products and ideas. So anyways, let me skip through this. So anyways, just to say, you know, again, we did through an academic process, you know, publish a bunch of papers around this work. And, you know, this is a particular class I've taught for a number of years, engaging mostly folks that had no bio background, actually. That's one of my things that I enjoyed most deeply, actually, is bringing folks that have no expertise, have never been in a wet lab before, and then giving them some basic skills so that they can actually start creating and working. Okay, I'll mention very briefly too, you know, we've talked a little bit about microfluidics. From an infrastructure perspective, one of the things that we built and that, you know, we published a Nature paper on a couple of years ago was this thing called Metafluidics. So again, I think in class you may have encountered other types of infrastructure, but this was our attempt to try to make a system where users could basically contribute different types of design files. So when I was in graduate school and I was doing work making these chip-based devices, it was so frustrating because you would go to the publication and there would be no way to get the design file. So I would literally be like looking at a little tiny picture in a journal, usually behind a paywall, and then try to figure out how did this guy or gal like make this thing. And so we built this site as a way to basically enable the larger community to share their design files so that now you could actually work from somebody else's previous work and you could also remix it, right? And that's one of the really things that we were really trying to get the field to go on, because you know, there's been tens of thousands of devices that have been made, but the vast majority of them do not get used, right? They get published one time, but then nobody else actually takes them and then does anything new with them. The very rare exception has a device that actually enables somebody to actually go remix it. And usually because it's a primitive that's simple enough to actually make yourself. And so what we've been trying to do with this site is try to enable more of that proliferation, which I think has been happening reasonably well over the past couple of years. So I give this as just one piece of infrastructure as an example. Okay, so I'm going to shift into other examples of kind of other big infrastructure projects that I think are open source and pretty cool. I put this up here just to say that, you know, this field of synthetic biology that we're in, and, I've given you a lot of shiny examples, but in many ways, the field is still very, very early. And I think is probably most analogous to like the era of early flying machines where people were just trying all kinds of crazy stuff, right? And, you contrast that with where we are now with modern aviation where, you literally can build an airplane like one time and it works. Like it flies out and it will just work after it's constructed. And, biology is just not like that, right? Like we are still figuring out what the first principles are. And so there's so much failure that's built into the life sciences and so much failure in terms of just the experiments and the things you're trying to test out. So it's just early days. And, the infrastructure is also very early, right? We don't have the equivalent of, the internet or personal computers for biology, right? It's not still so easy where you can actually just go and execute experiments. But there's a bunch of infrastructure that's being built now that may help, which I will get into now. So one really important entity is called the BioBricks Foundation. So the BioBricks Foundation was founded by Drew Endy, again, who is one of the real fathers of the field. And Drew, I think, I kind of like to describe him, I don't know if he would like this, but I like to think of him as kind of like the Steve Wozniak of synthetic biology. Like he was the guy that was like, we want to make BioBricks, we wanted this to be open source, we want the parts to be out there, we want everybody to be able to build with things. And he was a very, very open source driven guy. And so the BioBricks Foundation also is the entity that's hosted many, many important things. iGEM, which is the International Genetically Engineered Machines Competition, which I'll talk about in a moment, that was originally a project of the BioBricks Foundation before it became its own foundation. And the BioBricks Foundation is also doing things like creating this, which is BioNet. So BioNet is an example of a network, essentially, that can enable the transfer of physical biological materials, right? So we still do not yet live in the era where it's so easy to like print like a DNA plasmid that I could just do it myself at my bench. Now I can print very small molecules of DNA, maybe hundreds of bases or thousands of bases with some difficulty. And so BioNet is set up as an infrastructure so that if Eric's got a plasmid in his freezer that I need, that I'm looking for, I could actually easily find out and know that Eric's got the plasmid and Harold's got the plasmid. And then ideally the material transfer agreements, excuse me, are set up in a way such that our institutions can rapidly share those physical materials. Okay, so this project is sort of a policy project. It's a hardware and tracking materials project, and then also a kind of a larger network project.
Yeah. Can I interject again? I'm sorry, David.
Yes, please. No, of course.
Just for the students, right? The idea is that what David is doing here is building a structure for shared building of innovations by users and by the community and so on.
Yes.
So it's not necessarily that you would worry about the specifics, but if you were in any other area that's important to you, electronics or stargazing or anything else, what you see here is a model. It's like, what the hell do you do? Well, what David is saying is you create a bunch of tools that are tractable and cheap, right? And now he's moved on to sort of say, and here's how you can exchange information.
Yes, yes.
All of these are basic affordances of a user system that's sort of out in the community.
Yes, that's a great articulation. So, so both Metafluidics, BioNet, it's a coordination problem, right? It's like, how are you disseminating both information and knowledge, which I'll talk a little bit about in a moment, but how do you distribute and get out there fundamental aspects of what makes the field work? Things like design files so that you can actually go make the tools yourself. I don't have a slide on this, but many of these databases that I was talking about, if you Google parts registry, genetic parts registry,
right,
there's a whole bunch of these open source registries that have DNA sequences in there. There's a protein database that's out there that is widely used. AdGene is another bit of infrastructure where people can get
this information. So if I can interrupt again, David.
Yeah.
Sorry.
No, don't, please,
okay.
Sarah, go.
So we're halfway through, right, the talk time.
Yeah.
And what everybody wants to do is sort of abstract for themselves. Well, I'm interested in something else, right?
Yep.
What are the underlying principles? And so here what you're
Yes.
saying, right, is, well, you give them individual tools and then you give them a platform to exchange, right?
Yes. Yes.
And then you should go on, I think, to
Yes.
stay out of the details of it, but that's what this is about.
Okay, so let me jump ahead. Let me skip through some of this stuff. Oh, is this okay? Um, I guess one thing I'll briefly mention is just the idea of a cloud lab, because I think this is pretty cool.
Yeah.
You know, so if you think about the whole notion of centralized and decentralized, one thing that people are thinking about is, well, what if there were one big central facility that is making all, that's running all the experiments, and then maybe we don't need to have labs everywhere. Maybe there's like one or a couple of these big labs, but then you can then get shares of that machine time. So you can basically design experiments and run them in that using that infrastructure. So it's another modality to think about. In this case for the life sciences, how you might execute experiments without having to have it physically right with you, right? Which we've seen in computation, of course. Okay, so I'll jump now into just stuff about the spaces, right? So this is some just stuff about diversity. I think you guys know all this. We were talking earlier about Drew Endy and folks, but this is, I think, where you know, it gets interesting, right? So DIY Bio, as I mentioned, founded in 2008. And one of the funny things about it, actually, the very first meetup, it's a global movement now. Do you know where the very first meetup for DIY Bio was?
No.
The Asgard in Cambridge. Do you believe that?
No.
It's like the worst place you can imagine for like such a historic meeting, but it happened. there's a really funny group picture, but anyways, you know, this is one of the pictures that I really liked a lot, and I think, you know, maybe will resonate with this group. So this is Rob Carlson, shown as a cartoon here, who's one of the guys that really studies the bioeconomy. And he was quoted, this is in 2005 already, the era of garage biology is upon us, want to participate, right? So one of the big things in our field, right, in biology is that you need infrastructure, you need physical space, right? So this is, again, if you're doing electronics, maybe you can get away with doing stuff on a desk and you're okay. But in general, with biology, there's a lot of tools, there's a lot of infrastructure that you need. And I love this picture because it kind of shows, you know, he's got his autoclave and his PCR machine, and then he's got his like laundry machine, his washer dryer in the background, you know. But it's a pretty effective home lab, right? And much like I showed you earlier before with the high school students that built kind of that set of open source tools in their lab. So one big kind of abstraction is just like, what are the tools and infrastructure you need in your space of innovation, right? Whatever it is, right? For us in life sciences, we need physical space, we need tools. So how do you share that and actually, you know, make that more accessible? And so the community labs have been one of the biggest ways. So this is, you know, GenSpace I mentioned, which is in Brooklyn, BioCurious in Sunnyvale. And here, you know, I'll talk a little bit about some of the projects, okay, just to give you a sense of what's happening there. So another really important kind of principle I would say is, what are the incentives involved in the users, right? I'm not sure how much you've covered this in class, but, you know, why are people there, right? I think Harold or one of the other students was asking about this. What's the motivation behind people being there, right? A lot of it is curiosity. But for some folks, you know, they have some real projects they want to execute, but they can't get it done because they're not in a traditional academic lab or a traditional industry lab. So these are some examples of projects that this top one is called Real Vegan Cheese, right? So earlier on in the talk when I was talking about cellular agriculture, so like making meat products and animal products without animals, the idea for this was first shown actually in a community lab. So in BioCurious, this is like almost 50, like I want to say what year it was, almost 10 years ago, one of their first projects was, can you actually make cheese without cows, right? And so it was almost more of a speculative art project there, but they went at it. They were like, all right, could we actually engineer a bacteria to actually secrete the proteins that are in milk, right, and make cheese out of it? And so it was a really cool idea that now has become a real commercial thing. But I think part of what's really interesting about these community labs is that people are coming in not necessarily because they're trying to get tenure. They're not trying to publish papers. They're also not trying to maximize profits for the next quarter. So in my mind, and again, I think it would be interesting to study this more from a sociological perspective, but I think you end up with some, in a way, like wilder ideas. Like there's a lot kind of crazier stuff that happens in these spaces because people aren't necessarily guided by a lot of the same incentive structures that you might see in academia or in industry. And so, you know, I've mentioned to you before, you know, some of these other tools, these kind of desktop biolab tools, which again, you know, these weren't ideas that were really being explored in academia or industry. It was really the community labs were the ones that were thinking to themselves, you know, how do we build a commodity liquid handling robot or a commodity desktop lab? And so the other kind of exploration I think that's been really cool is art, right? So the open source gender codes was a project out of Bugs Lab in Maryland, where you basically had a bunch of trans activists that were looking at hormone production and said, hey, what if we think about synthetic biology as a way to reclaim hormones and thinking about how do we manufacture them for ourselves? So in a way, it was sort of this project was more of a bio art project, but it was a cool one in that it kind of got you thinking about, hey, like how could I with the life sciences actually make my own hormones, right? And similarly, Glowing Plant, which was a kind of controversial project at the time, which is now, MIT has got big research programs on it, but this started as a Kickstarter in BioCurious, right? Could you actually make a glowing plant, right? This was years before MIT and other universities started picking them up. So part of what I'm trying to say is there's been, I think, a real amazing kind of explosion of different cool ideas that you see in these spaces that you might not see in a traditional spaces. So where are these labs happening? There's labs being set up in public libraries. There's the BioBus. I think Jose Gomez Marquez, who's a dear friend of both myself and Eric, they're trying to figure out how to do maker spaces inside hospitals. This is one of the coolest ones that one of my former students has been working on. It's a floating biofab lab. The vision for this thing is a biolab that actually floats down the Amazon River, can do bio experiments and engage with indigenous communities as it goes. And they've been working on this for a number of years, and I think it's just such a marvelous example. And again, these are not academic or industry labs. These are just folks trying to figure out how to build labs in some of the weirdest, coolest spaces that that I've seen. So just shifting gears a little bit into the education part. One of the biggest infrastructures that I think has been set up is the International Genetically Engineered Machines Competition. It's called iGen. It's basically a big synthetic biology nerd competition. Thousands of students from all around the world participate. It was hatched in a way to propagate this idea that biology could be set up into reproducible parts. So all of the students have to work with these BioBrick parts and then build something cool genetically engineered something from these parts. So that was kind of the basic kind of modality going into it. And now the idea was to kind of bootstrap the parts library through this. Was it successful? I would say right now, if you look at the parts that are in there, there's still a very small number of parts that people actually use. So the aspiration was always to get towards like the engineering design library, right? You've got these very robust set of parts and you can pull from them and then build whatever you want. It turns out it's not quite so easy. But I think it's led to the evolution of a number of other part libraries that have become a little bit more robust and used in industry. And so anyways, I'll just say I'm iGEM's DJ. It's one of my favorite roles over the years. And then, in our own community lab here in the Boston area, which Eric has been to, this is I'll just briefly tell you this and then we can shift gears, Eric. But in our space, it used to be a Chinese language bookstore called East Meets West Bookstore on Mass Ave in Cambridge. Many of you may have walked by it. My father, as I mentioned, was a professor in electrical engineering and computer scientist in computer science. So his three favorite letters were EMW for electromagnetic wave, which was his area of expertise. So we called the space EMW. And, we started out as like a poetry space. It was a space for like poetry and open mics and, worked with marginalized communities. I mentioned I was a social justice activist. So, we worked with, these are just some photographs of our team, but, poets and artists and electronic music producers and all of these wonderful creatives, jazz musicians and so on, beatboxers, visual artists. But what we did was, right, we eventually built out our own community lab. So this is a picture of that. So we started doing our own biohacking experiments in around 2014 or 15. And so this is us basically doing a process where we were synthesizing DNA, putting it into organisms. And this was with a room full of artists and designers, grad students also from the Media Lab, who had never done any wet lab biology before, right? So to me, this is always a thrill. This is Professor Neri Oxman, who had never done any wet lab bio before at that time. And over the years, we built out a whole bunch of really cool programs. And so this is, again, I mentioned Ginkgo Bioworks, we got all of our early equipment from there. And, to us, a huge part of it, again, was the community infrastructure, right? So getting together as a big group, learning from each other. And I would say, Eric, I think one of the parts that makes a lot of the community labs maybe more successful is that people are coming in with actually similar levels of understanding, right? I think the people that come in that have more technical expertise oftentimes know that they're the technical expert, so that they know that, hey, in this room, I'm the teacher, right? So I'm here to actually help and instruct and provide more knowledge and understanding. The technical experts then often can then, if they have some of these labs have a member model where you'll then actually can rent a bench space for very, very low cost. Those folks can then be doing their own projects, their own innovation projects. So there's ways inside these spaces to kind of have a hybrid of modalities between kind of learning and innovation, which I think it may be useful to point out.
Yeah, great.
And so, anyways, over the years, all kinds of wonderful workshops, art galleries we've done exploring the interface between citizen science and bioart. I think one of the coolest things we've done over the years, going back to education and learning, is engaging with young people. So, we've been active participants in the Cambridge Science Festival every year, getting them excited about bio and the life sciences. This is a program that we had called the Youth Science Initiative, when we brought our students to places like Ginkgo Bioworks, to the public library, to the science museum, working with astronauts and so on. So again, and I think these are just examples of activities that I think are pretty exemplary of, or pretty typical, I think, of many of the community labs that are all around the world. So, but now I want to bridge between education and learning and innovation, right? So there is actually, I think, a very strong intersection. So there's a class that I've been teaching for the past 5 years now with Professor George Church called How to Grow Almost Anything, okay? This is a class that's designed for people that do not have any bio experience, but basically the kind of teaching philosophy is every single week, we introduce you to one topic at the cutting edge. It could be 3D biofabrication, it could be biological production, or ecosystem engineering, whatever it is. And the goal of each class is to give you just enough skills that you can have a hello world type of demonstration within that class, okay? So we really try to structure it that way. And ideally, the skills build on each other over time, so at the end of the class, you can, quote unquote, grow almost anything, okay? It turns out, from an educator perspective, it is extremely challenging, especially in the life sciences, to design a class like that well. Partially because week by week, just biology is just slower than electronics, right? You don't have to wait for your electronics to grow in a petri dish where you do in bio. So there's just a different kind of time constant there. But we've learned a lot over the years. And what was really cool about this class was we started it as a global curriculum, okay? Some of you may be aware of what's called the Fab Lab network. I'm not sure have you've heard of this, but it's basically this global network of these laboratories that have digital fabrication tools, so like laser cutters, water jet cutters, 3D printers, etc. This was structured by Professor Neil Gershenfeld, my colleague at the Media Lab. And Neil basically built out this global network of these labs with a common set of tools, which again, another thing I think you may have talked about in class is common tool sets. If you have common tool sets, then it makes it way easier when you're at a distance to still kind of work on similar projects. And so what George and I were basically tasked to do by Neil was, can we actually get these fab labs to basically build their own biolabs, right? If you've got a fab lab, maybe you can actually manufacture all the tools that you need to actually have your own biolab. So that was kind of what we started working on. And so this was one of the very first kind of fab conferences where we started engaging this maker community to get them into bio. And over the years, again, we've had this really wonderful experience of just working with these labs all around the world, getting them this curriculum and getting them building and making bio. So these are just some pictures in our lab. And for me, it was like, this is my parents' Chinese language bookstore, so it's like, I never in a million years would have imagined that, like, whatever, 20 years later, I'd be teaching like a cutting edge global synthetic biology class with George Church in my Chinese language bookstore. You know what I mean? It's just life is such a weird thing, and it unfolds in such unexpected ways. Yeah. And so, yeah, really cool projects, biosensitive tattoos, living color palettes, and so on. So really, really cool projects. And so the reason I mentioned this as an intersection between the learning and education is we're teaching a curriculum, but it's all project-based, right? Where the whole goal is to actually have it be experiential, so that the students that are in laboratories around the world are actually hacking and building and getting that hands-on experience themselves, right? So, a real project-based learning. So hopefully by the end of the course, you've got that kind of basic skill set, so that, and now you've got a sense of the tools, right? Which we've talked a lot about. How do you kind of get these tools out there? And this is very much active research for us, right? We still don't know exactly what the perfect tool sets are and what the best ways are to get them out there, because our global network, I mean, we literally have labs that are in like Kumasi, Ghana that, you know, if their thermocycler breaks, nobody is coming to fix that thermocycler. You know what I mean? They're not going to ship that thing to Germany. So they need to learn how to fix it themselves. So we've really been trying to figure out how to get the tool sets, make them as open source as possible so that people can hack and build with them themselves. And, you know, maybe one of the concepts that, Eric, you might like this, there's something called biosafety levels, right? So the biosafety level 4 is like anthrax, you know, biosafety level 1 is like typically what's at MIT and, you know, high school laboratories. George and I have been thinking a lot about what's biosafety level 0, right? What's biosafety level 0? Like literally something that I could work on, you know, as I'm talking to you guys right here in my desk, you know, in my office at home, you know, something I could do there. So anyways, so this is, maybe I'll pause here for a moment. There's one final kind of category of stuff that is more about the global network and the BioSummit. That's the final area. So Eric, I can go into that now, or we could pause here if you like, whatever you'd like me to do.
Yeah. Well, I always think it's nice if we can have people sort of think themselves a little bit in terms of small groups.
Great.
So let me try something out on you, David. I mean, David is amazing in the sense that he's a force of nature. I'm always very impressed by David. And what he's also very collaborative, and what they're doing is they are building sort of this area into an area where the hacking is easy enough so people can enter, where there's equipment. It's a big infrastructure thing. But now, so maybe what we could think about, because you're all interested in different areas, and so maybe what we could think about in a breakout for say 12 minutes is, in my area, whatever my area is, you know, whether it's medicine or whether it's manufacturing or whatever the hell it is, or how could I create a tool set for users to innovate on their own, right? Everywhere you go, as in, you know, especially Sloan students and engineering students too, you go into a structure, you go into a setting where all the stuff is around, and here's how we do things around here. Now what you want to do is rethink, just as David is doing in the area of bio. Well, how the hell can I democratize this? How can I democratize whatever the heck my company does?
Well, why don't we take a few minutes? I would love to hear, I mean, I know in our discussion, we had a really nice discussion thinking about barriers to entry, which I thought was a really nice way to kind of frame it. Like if you wanted to imagine whatever your discipline was, you know, one way to think about it is what are the obstacles, right? So in our group, we were talking about, you know, cost as an obstacle, right? You know, and in various industries, how do you bring that down? And then, you know, we were talking just about, I think one, you know, Georgia, I think was, had her area is thinking a lot about farming and agriculture. And I think a lot of the issues there are quite similar to biotech, right? In a world of where matter is important and physical stuff is important, you know, just getting the physical stuff and moving it around is like really can be very complicated and very hard. You know, that was sort of like the BioNet program that I mentioned. And, you know, even connected to pandemic response, right? You know, we're facing so many supply chain issues. And wouldn't that be mitigated if we had more decentralized, you know, manufacturing of like reagents and other things as well? So from a pandemic preparedness perspective, I know we're doing a lot of work, you know, with pharma and other entities to try to figure out for future pandemics how to decentralize and make these supply chains more robust. So
yeah, fabulous, fabulous. So anybody else?
Eric and David may actually nominate someone from our group.
Of course. Mm-hmm.
We had
Please.
an amazing example from Yena, and Yena, if you don't mind, maybe you can go live and share it with others.
Sure. So I'm a surgeon. I have a private practice as well. And one thing that I'm finding very difficult in practicing medicine, especially in this day and age with the pandemic, is wearing our surgical mask or N95 mask. I find it's very uncomfortable because of our face type. I have a longer nose and a wider face. And I think also with my colleagues, I'm hearing a lot of complaints of the fit. Is it fitting us optimally? Is it protecting us? And then we always walk out with the gouges around our nose and our cheeks and so forth. And toolkitting and being able to look at different types of face structures and being able to really find the perfect fit for the surgeon or the doctor or the nurse or whoever's taking care of people, to be able to do that would be so ideal. And the other issue that I'm running into, as everyone knows, is the lack of PPE. I had procured some masks from China, but the supply chain has really been curtailed due to the FDA stepping in and wanting to make sure, which is something that we need to do anyways, that we're not getting substandard masks and not protecting our medical workers. So I took the next step of then thinking and figuring maybe possibly making our own PPE here. So I studied the materials that we typically use, and that's polyester, it's non-woven material, melt-blown material, and looked into how they're created, and that's with an ultrasonic welding machine. And I purchased one. And now I'm looking to try to find some of the materials, which I'm running into roadblocks because most of them have already been spoken for because this is such a pandemic. It's
Yeah, that's good. That's good. Thank you. So can I suggest to you, David, that we better, you have less than half an hour left?
Yeah.
Yeah.
Sure.
Thank you.
So, yeah, thank you. This really, I love that so much. So I can wrap up with just a little bit of discussion about kind of what we're doing with the global network, and then we can just wrap up with some concluding discussion and remarks. So let me try to share my screen again, and hopefully this all works. Okay, are you seeing my my screen?
Yeah. Yeah.
Okay, great. So, so again, I joined the Media Lab. You know, came back to the Media Lab about 3 years ago to start this group. And basically, you know, I started going around the world visiting these different spaces. And so, you know, I met all of these amazing colleagues. This is Thomas from Cameroon. This is Haquarium in Switzerland. And again, I think this is a worthy topic for this group too, thinking about security and ethics. You know, we talked a little bit about that earlier, and I'll talk about that more in the end. You know, again, we have a very close relationship, interestingly, with the FBI, at least in the United States. And so I kind of, you know, I'm from Lexington, Massachusetts, so I was doing my sort of Paul Revere thing of kind of going around the world and trying to visit these labs and understand what's going on. So these are this is a really big conference that I went to called BioFabbing Convergence in 2017, which is this big kind of a conference for the DIY bio and biohackers in Europe. And then again, you know, all throughout Asia, for myself, I was visiting different spaces. These are, I'm a photographer as well, by the way. So these are some of my photographs. So this was, you know, this is Georg from Japan and all of these wonderful folks, Bio Club Tokyo. And so this basically led to the Bio Summit, right? So in 2017, when I joined the Media Lab, you know, the first thing we did was to pull together this gathering, which was, you know, it was basically like the family reunion with the family you didn't know that you had. You know, it was like people finding their tribe. And so again, you know, I'm trying to map this and think about, again, from principles to other spaces. But to me, the actual things like conferences and events where you can actually bring people together, ideally in person for that human connection, I think is just so valuable. And something that, you know, when we got this group together, you realize that even though we're all physically separated, we have so much alignment in terms of our values, in terms of our mission and what we want to achieve in the world. And so the first year we had, you know, a couple hundred participants from labs around the world. We thought, were 50 people going to show up? We don't know. But one of the most inspiring things to see was just this incredible diversity of both geographic locations, perspectives, and so on. And I think one of the real strategic things that we did, and again, I'd be very curious from Eric and others, you know, how well you might consider this or map this to your discipline. But one thing, you know, I mentioned I'm a social justice activist and a community organizer by training. And so one of the heroes that I have from community organizing is this guy, Marshall Gans, who some of you may know. He's at Harvard at the Kennedy School. And, you know, Marshall is a legendary community organizer. He literally was an undergrad at Harvard and dropped out to move to Mississippi for the civil rights movement in the 60s. So, you know, he worked with Martin Luther King, Cesar Chavez. He organized Obama's campaign in 2008 and 2012. And so what I did was very intentionally injecting this idea of social movement into this larger kind of scientific nerd community, right? So you had a bunch of nerds that are like, I'm building labs. And what we got them to think about was, you're actually part of a larger movement. There's another framework to think about this, which is actually movements. And that there's actually skills and strategies and methodologies whereby you can actually deploy a movement, right? And so the first year of the BioSummit, one of the kind of questions, one of the framing questions was, are we a movement, right? Is this a bigger thing? Is this some kind of a larger social structure through which all of our individual community labs that we're organizing are a part of? And so in the successive years of the BioSummit, I think that framing of a movement, I think, has been very, very valuable, and it's allowed us to do some, I think, really interesting things, which I'll talk about now. So BioSummit, of course, we're not going to have an in-person one this year. It's going to be a virtual event, but we're still going to keep it experiential. Okay, so BioSummit, much like how to grow almost anything, much like all the stuff that I do, it's all about the experience. So we have all of these hands-on workshops, and so literally, our labs are kind of bursting at the seams at the Media Lab, and all the wet labs that we get activated, and people are learning all kinds of stuff. I mentioned I'm a DJ, so we always have a fun, awesome party. And so, the first one was this kind of historic, like, family reunion of the family you didn't know you had. The second year of BioSummit, in 2018, we almost doubled in size, and in terms of the participants that could come. And, again, I think, we really started to see a lot more emergence also in particular from the global South. So Latin America, Africa, really amazing laboratories are being set up there in response to all of this work that we've been doing collectively. And this is where I really started to advance this idea of this phrase of community bio, that maybe this is like an evolution from do-it-yourself biology. And so here, again, I started collaborating around this time with one of Eric's colleagues at Sloan School, Professor Thomas Malone. So Tom and I think, really started to get along great, because he'd been thinking about, how do you structure basically communities, right? Or different types of what he calls superminds. Of course, I'm really interested in communities. And so we got together to ask, well, okay, what are the ways that we could structure these communities, and perhaps through that structuring enable more innovation, enable more learning, right? And I think that's one of the big active questions that we're still trying to grapple with. So, again, Tom thinks a lot about collective intelligence, which is something I've been learning a lot about. But fundamentally, right, I think that this may resonate with some of you. This movement, it's like a bunch of pirate ships, right? There's no CEO, there's no captain that says, like, even me, I think I've got a lot of soft power as a leader, and people look up to me in a certain way. But I can say all I want. Hey, GenSpace, go do this. BioCurious, go do that. Josiah Zyner, go do this. They're not going to listen to me. They're going to do whatever they want, right? It's not a company, right? There is no structure where there's a CEO or a dictator or even a president, right? So how do you get like a big disparate fleet of pirate ships organized, right? Which I think is a really interesting question. And so here I go back to movements, right? So what brings people together? What brings movements together even? So a shared sense of values, a shared purpose, right? A shared set of norms, a shared set of ethics. So these are some of the things that I think are often part of movement building practice. And so what we did at the second BioSummit was we tried to make explicit some of these implicit things. So we did work to try to articulate what are the values of our community, right? So Gunther Weil, who's a very dear friend and collaborator, that's done a lot of really wonderful work looking at values and had whole systems around thinking about how you analyze and structure values. So we went through some exercises, looked at this for our community, which I thought was really fascinating. We did a first set of charrettes around ethics. I'll talk about more in depth what we did which we did in last year's BioSummit. But again, critically important, right? If we want to have a lot of innovation in the life sciences, which we desperately need, how do you do it safely? How do you make sure that we're creating the right types of culture and ethical frameworks for that, right? And then the other big thing that we did was around vision. Okay, so this is one of the cooler things that we did. So again, working with Marshall, we went through a process whereby we first started with an initial group of core organizers to generate what we call the statement of shared purpose, right? The idea here is, is there kind of a larger kind of framing that we can all rally behind and agree to that, hey, we're all here and we all think that we're a part of this larger community bio movement because we have this shared purpose. And I think it was a really important exercise for us to articulate what that purpose was. And so we went through an iterative process of first working, first the organizers, then at the BioSummit itself, groups of 50 to 75 people. And then finally, with the whole conference, with hundreds of people, we worked on generating and synthesizing this statement of shared purpose. And I'll read it to you because I love it so much. And we call it version 3.0 because these are versioned, right? The whole point is that these things change over time because we all change over time. So it reads, Our shared purpose is to fundamentally transform the life sciences and democratize biotechnology to inspire creativity and improve lives by organizing life science changemakers and bioenthusiasts to build an inclusive global network, cultivate an accessible commons of knowledge and resources, launch community labs and projects, and enable local educators. I don't know about you all, but like when we unveiled that in kind of the closing session of the Bio Summit, I mean, people got up and they were applauding. I mean, it was so cool. And the other part that was so cool is the co-creation part, right? People could see like, oh, like our group contributed the thing about, the accessible commons of knowledge, right? So I think there's something very, very powerful about our group all working collectively to actually shape something that we all felt and believed for ourselves. So, and this is, again, really out of the Marshall Gans, movement playbook, which I thought was a really interesting combination. We've got a hand raised from Beshimov at MIT. Go for it.
Thank you, David. This is me here. Your comment about your soft power and your earlier comment about Steve Wozniak
yes.
sparked the question. When we look at Steve Wozniak, we're thinking about this tension that existed between open architectures espoused by him and closed architectures espoused by Jobs.
Right.
And I'm sure some variant of this tension is being played out in your community. There are
yes.
members of your global community who espouse closed architectures.
Yes.
And it looks like you're espousing an open architecture. Can you speak to that? Is there a tension like that?
What
is happening?
Yeah, for sure, for sure. I mean, I think in general, and again, so many of my very dear friends and colleagues have done, I think, an amazing job of basically industrializing synthetic biology, right? So, all of these companies, Ginkgo Bioworks, these billions of dollars that are getting pumped into this industry now. I mean, there's a very successful synthetic biology industry now that exists and has matured, and huge infrastructure built to support it. Giant conferences, consortia that have been built out, all kinds of VCs that fund the work, and so on. So I would say the industrialization has been very successful. And interestingly, I think somebody like Drew, we've had a number of really interesting conversations more recently talking about things, because I think those of us that really are thinking a lot about the open source parts about it, the user-driven innovation parts, the open innovation parts. It's not that we've been sidelined per se, but I think it's just something that's co-evolving at the same time. And part of what's interesting too, even Ginkgo, right? Ginkgo, the founders of Ginkgo Bioworks were also the founders of Open Wetware and iGen. So there's actually a lot of very strong value alignment between industry and kind of these community infrastructure. But the question is, how do they talk to each other, right? So Ginkgo, and I work very closely with them on technical projects and so on. They're always trying to figure out, like, how do we balance our, in a way, just institutional obligations as a for-profit company with things like they're sitting on top of the single biggest database of basically kind of knowledge on how organisms react to different types of DNA programs. Like Ginkgo has, it's called Codebase. They have something called Ginkgo Codebase, which is literally their repository of all of their knowledge of all of the experiments that they've run. And because of all the laboratory automation and everything else, that code base is probably one of the most valuable pieces of kind of bio knowledge that exists, right? And so there's aspects of it that are very proprietary that they want to keep for themselves, but they're also very interested in figuring out how to support the larger ecosystem in the community. So I would say, there's just a very interesting dynamics that are at play there. And, I think a lot of it comes down to institutional incentives, right? What are the outputs that are driving, certain types of institutions? So yeah, I don't know, Eric, if you want to chime in here too, but it's something that I've seen that is absolutely playing out. And
yeah, well, I just want to point out to the group that what we have is well-worn paths, for instance, in the business school. Well, what you can do here is you can commercialize this. And it becomes sort of the default. And as David was mentioning, a lot of the things that sort of started in the open in a way, sort of now people have founded companies on them and so on. But the marvelous thing that I think David is doing, sort of almost as an organizer, amazing guy, is saying, well, how do we build the rules for an open, inclusive network, right, that is viable? I mean, that's the key, that's viable. So I would like to know from you, David,
Yeah.
what's viable about this? What is your impression, right? What's
Yeah.
your sense of viability?
Such a great question. And this is something we talk a lot about as a global community, right? I
Mhm.
actually think that one of the biggest questions for us actually is a business question, frankly, which is a business model question, right? How do we, as a global network of laboratories, ultimately sustain ourselves, right? What I found so fascinating is in the United States, the vast majority of labs that have been set up, there's a hobbyist element to them, and that people have full-time jobs doing, whatever they're doing, and then they come to the lab on the weekend or they support it and so on. And many of these labs, most of the labs in the US are not for profit, right? They're nonprofits. And so they're getting their funding from grants. Some of them also have membership models. Many of them have membership models, which actually is quite successful. So BioCurious, which is the one out in Silicon Valley, I mean, their lab is just exploding. I mean, it's crazy. And, they do open community projects that are educational, and like high school students are coming in and learning about all kinds of crazy stuff. But then very serious companies that have now become very serious companies started there because they've got the cheapest bench space, right? They're not, set up in the same way as like, Berkeley Biolabs or that, kind of startup accelerator is. And so, I mean, it's actually amazing. Mammoth Biosciences, which is on the cusp of producing one of the kind of premier CRISPR diagnostics for COVID, which potentially could radically change the game, started at BioCurious, right? So they got their first bench space there and they got started there. So, but the question in terms of viability is, BioCurious has figured out a model that works in part because of their proximity to Silicon Valley, right? I think they've gotten something that works well because they can do workshops. Like when Samsung is like, what is going on with CRISPR? BioCurious could say, hey, for $20,000, like we'll take your executive team and show you what CRISPR is, right? So there are ways that, oh, I'm not sure what's going on. Okay, but one of the big questions I think is just a business model question, right? And so for the hobbyist scene, maybe it's a little less, we still kind of keep it at that level. But in the global South, right? I have colleagues that are setting up labs in Africa or in Latin America in certain places, and they're trying to build bioeconomies. Like they're trying to figure out how to turn this into like a full-on profession, right? And how to actually attract the capital and really turn it into from something that may start out hobbyist and open, but actually has a working business model. And actually, I would invite you all to come to BioSummit 4.0, which is going to be in October of this year, because this is one of the recurring themes that we always talk about is sustainability, right? Mm-hmm. And again, when we look at open source, right, I think there are some marvelous examples, of course, of institutions that have become very financially stable and have managed to incorporate the open in with the closed. And again, I think this is, Eric, you're really legendary at a lot of this work. And so for me, I think that's one of the big questions going forward. And, I've got colleagues that are thinking of some crazy ideas. I mean, like, is there a way to pull blockchain into this whole thing? So now you've got, kind of a way to basically leverage the decentralized nature of that to enable certain types of decentralized work and business models around that. But I think that's one of the big questions, Eric, that we're really very keen to come up with better solutions for.
Yeah. So the thing is that what David does, most people in most of these organizations are inventors of some sort. That is, they like the technology. David and some of his colleagues really like the social invention too. And I think it's very important because at the moment what happens, I mean, the traditional example has been, what did you call that thing, that sort of group in Silicon Valley that collapsed when the computer, remember there was an infrastructure, Steve Jobs and everybody else was sort of operating non-commercial.
The hobbyist, I know exactly what you mean. Yeah, that
was
hobbyist
a
network.
Club, and everybody was sharing. And then all of a sudden, somebody thought, whoa, we could start to make money from this. And the whole thing, the commons blew up.
Right.
Now, an interesting contrast that David mentioned is the licensing model in open source software.
Yes.
Where it really prevents sort of everything being blown up and turned into commercial private organizations. Now, I think there's an interesting model there to answer your question, David, because the point is that what you need is, when you've got this collaborative thing working out really well, like Linux and the rest of it,
yeah,
it is more powerful than the commercial system.
Yes. Yeah.
And so what you've got to do is set up a system that basically can protect itself, you know,
Mm,
can sort of share so that people can, right? I mean, nowadays proprietary software is dying and open source is dominating. And so I think for your thinking as a political figure in this, you know, that's the kind of model you want. Something
yeah.
that doesn't depend on charity. It depends on the incentives of the actual innovators themselves, and sort of getting corporate incentives aligned to that. Because nowadays corporations all want this basic stuff to be open. You
Right.
know, they don't want to do that.
Right.
So yeah, we can talk about that more. But to the rest of you, what we've really what's so exciting about this is that we have a well-established model historically in business schools and everywhere about, oh, well, what you do with all this stuff is you start a company and you divide up the commons, and you know, then you've got all these patents, and progress is slowed down in many ways. So what's being built here, and what I'm so proud, if David allow me to be proud of him, what I'm so proud of David about with his colleagues is that they're trying to think about the infrastructure in a systematic way, because there's really no reason why a lot of this couldn't be designed in a much more sort of open way to everybody's commercial benefit, as well as, you know, as well as open source sharing benefit. So that was my rant, David, sorry.
No, Eric, thank you so much for saying that. No, and again, I think just, you know, selfishly, I think even the past couple of weeks, like, dude, we got to hang out more, man. This is like, we like, really, I think like so much of what you're talking about and what the students are learning about in the class, I think it's so important, because, you know, I felt the exact same thing. Like, even when I was in grad school, it's sort of like, okay, well, I want to do innovation, but it felt like the only viable choice was to do a startup. And that didn't necessarily align with my own values or what I wanted to do. And I think, you know, trying to figure out and articulate these alternative structures is so critically important. And, you know, these are all experiments. I mean, we literally, we're building this and trying different things. And I think, you know, maybe the final thing I'll share as a part of this, you know, I just want to address the ethics question really quickly. You know, the last year's BioSummit that we had just in October, you know, we could barely all fit in the same room anymore. And, you know, it was such a beautiful thing. And you had more than 600 people that were accepted from all around the world that are, again, just a part of this really beautiful global phenomenon. And, you know, so this is something for BioSummit 4.0. We just had an organizing meeting today talking about how we can still have this experiential structure regionally while we do a big global event. So the event in October, I think it's going to have a feel like a 3-day, like 72-hour or 24-hour day hackathon. Because if you're sleeping here in Japan, they're going to be doing stuff, and in Singapore, they're going to be doing stuff. And so I think the way that we're going to structure it, it's still going to be virtual, but still so much experiential, hands-on stuff that's going to happen as a part of this, which I'm really pumped about. But, you know, when we think about that infrastructure and these different types of social structures, right? In my space, you've got a structure built around a lab, right? That's one core thing. And I think, you know, as Eric talked about, you're going to have somebody talking about maker spaces. And, you know, these physical spaces are still very important, because you get that kind of community interaction in the physical environment. But part of what we did between years 1 and 2 was a leadership thing. So we worked again with Marshall, and we built out a fellowship program where we identified 36 people from all around the world that, you know, we thought were either setting up labs or people that could be kind of accelerants in this larger movement building process. And basically, you know, again, it was an incredible group all across the world, very strong representation in the global South, in particular, Africa and Latin America. And we went through this whole like kind of 8-session leadership development process. And then critically, they all had global projects they were working on. So they all came to the BioSummit in October, but they've been working over the summer on these different projects. And so again, you know, thinking about ways to generate innovation, it was really neat for us to kind of have this combination of leadership, movement building, where it's like, we actually want you to engage in a campaign and come up with some cool projects. And they came up with amazing projects. So, one here, global exchange network, sort of like the BioNet version. How do you actually get physical materials around the world? We had one that was all about diagnostics and public health, right? So how do you ultimately, address this question of low-cost diagnostics, which now, of course, is such a massive deal in the world? How do you do things like share protocols? That's a huge, huge area. How did open versus closed protocols, et cetera. So anyways, all that to say, this was a really cool kind of thing that we did. And then the other, in closing, the big thing that we did was ethics, right? So we had a whole community-driven process where we again, did not know what the community was going to come up with at first, but we co-created a whole set of kind of ethical principles and guidelines that, again, I think I had not been a part of in an academic perspective, so it was really cool to see come together in this community one. So,
Yeah.
All right, I'll stop there.
So first of all, fantastic. And we haven't figured out a way to collaboratively clap yet. But I think everybody's as enthusiastic as I am. I mean, fabulous.
Oh, thumbs up. Thank you. Thank you. Appreciate that so much. Yeah.
Circuit figured out how to do it. Okay. So anyway, that's really good. And what is important for the students, basically, and all of us, is what you see going on here is sort of an infrastructure development and the principles of it, which are as rich potentially as this corporate culture that we are now based upon. Right now, it's sort of like there's this central way we do things through companies. They certainly have a value, as you've seen in our talks. There's this innovation process that is corporate, but there's also this other innovation process. And the fascinating thing to me is that you're all going to have to deal with it, and in a good way, because where it has been really fleshed out, like in open source software, it tends to outcompete companies. So it's a brand new way of organizing our economic and social interactions, and you're in an exciting time with respect to that. Well, that's my rant. Berdine, do you have a rant as well?
No, I do not. Well, I can rant very positively for a long time, but thank you. This was a marvelous session. I'm so grateful that I've had a chance to participate.
Yeah, yeah. All right, so it's 6 o'clock, and thanks to everybody, and we'll have our final class on Monday.
Yeah, and please feel free to send me email if you want to stay in touch or chat more. I'm very, very delighted to. I think for me, one of the great joys of being a part of a class like this is just interacting with folks that are not biologists and getting your perspectives. So I'm on all the social media stuff at David Sun Kong, and you can email me at dkong@MIT. So I'd love to stay in touch. And Eric, thank you so much again for having me and being a part of the class. Really, it's such an honor to be a part of it. So thank you so much again.
Fabulous. Okay, thanks to everybody, and we'll see you on Monday. And thank you, David.
Of course. Thank you all so much again. Take
Okay,
care.
bye.